The process of manufacturing food by chlorophyll in green plants is known as photosynthesis. Now, the reactants of photosynthesis are light energy, carbon dioxide, and water. And the end products are glucose or food, water, and oxygen. Now, science begins at home. Now, let us try out a few simple experiments to find out the authenticity of this entire process of photosynthesis. Now, have you seen plants like these that have leaves that have some part of it that is green and some part that is non-green? These leaves are known as variegated leaves. Put iodine on it. Now, iodine has a special feature. Iodine gives a blue-black coloration when it reacts with starch and remains its own color, which is a brown color when there is no starch. Now, look at this leaf. This part, which is green, will photosynthesize, thus forming starch. And this part, the whitish pink part, since it does not have chlorophyll, it won't photosynthesize, so no starch will be produced. So when you put iodine on it, only this part will turn blue-black, and this non-green part will remain brown. So from this simple experiment, we can conclude that without chlorophyll, there won't be any photosynthesis. A potted plant is kept in darkness for 48 hours. After 48 hours, it is taken out in the sunlight. One of the leaf is wrapped in foil so that it is not exposed to sunlight. And then both the leaves are cut from the plant. Let us see how he is doing this. The leaf which was exposed in sunlight was cut and kept in boiling water. And also the leaf which was wrapped in foil paper that was not exposed to sunlight was cut and put in the boiling water. Now, after being kept in the boiling water, next, take out the leaves and put it in test tubes. After putting it in test tubes, pour some alcohol in the test tubes. And place these test tubes in boiling water once again. Now, after some time, take out the test tubes, drain out the alcohol, and take the leaves and put it back in boiling water. Now, after that, pour some iodine solution on both the leaves. Now, this leaf, which was exposed in sunlight, immediately showed a blue-black coloration, whereas this leaf that was foil wrapped and was not exposed to sunlight, when iodine was applied, remained brown. Now let us recapitulate what we did in the entire experiment. First, we kept a potted plant for 42 hours in a dark chamber so that the plant gets destarched. Now, what do I mean by getting destarched? Well, if you keep a potted plant in darkness, then the leaves will not be able to photosynthesize anymore. So the amount of food that was already stored in the plant will be utilized completely by the plant in that time. So now, after 48 hours, there will not be any starch present in the leaves. Next, when the potted plant is kept outside in the light and only one leaf is foil wrapped, then these leaves which are exposed to sunlight will photosynthesize. But this leaf which is kept in the dark, that is wrapped by the foil, will not be able to photosynthesize. Next, we'll cut both the leaves. This leaf which was covered by the foil and a leaf which was exposed in the sunlight, and we had taken both the leaves and we had placed it in boiling water to kill the cells. Now, what is the purpose of killing the cells? Well, this helps in increasing the permeability of the cells to the iodine solution which we had later used. Now, after heat killing the leaves in boiling water, we had kept it in alcohol. Now, after keeping it in alcohol, we saw that the leaves became colorless. That is, there was removal of chlorophyll. Now, how is that helpful? Well, if the leaf becomes colorless, then the color changes during the iodine test will be more visible and distinct. Now, after taking out from the alcohol, the leaves become very fragile and brittle. So it is again kept in boiling water so that the leaf softens. After that, both the leaves are kept on a pallet and iodine solution is dropped. The leaf which was exposed in sunlight had photosynthesized, so it had produced starch, so it turned blue-black on the application of iodine. But this leaf which was wrapped under the foil did not photosynthesize, so it did not have any starch content in it, and so it remained brown on application of iodine. So from here, it is evident that starch is produced in the presence of light only. Now, let us perform a simple experiment to see if carbon dioxide is necessary for photosynthesis. For that, a potted plant is kept in darkness for 72 hours. Now, 2 beakers are plugged tightly and leaves are inserted into the beaker. One beaker contains sodium hydroxide and another beaker contains water. Now, this entire setup is kept in sunlight, and after a few hours, both the leaves are plucked and kept in a petri dish and then heat killed in boiling water. After that, it is placed in alcohol. Now, since the leaves become very brittle, it is again placed in boiling water, and then both the leaves are tested with iodine solution. The leaf which was in the beaker containing water turned blue-black, whereas the leaf which was in the beaker containing NaOH solution did not. Again, let us recapitulate the entire experiment. What did we do first? Well, first we destarched the plant by keeping the plant in darkness for 72 hours. By destarching, I mean there won't be any starch present in the leaves because all the starch would have been utilized by the plant during the 72 hours. Next, a beaker containing sodium hydroxide and another beaker containing water is tightly placed around 2 leaves and tightly corked with cotton balls. Now, what is the significance of this process? Well, sodium hydroxide absorbs carbon dioxide from the air and turns white. But water does not have the property of absorption of carbon dioxide. Next, both the leaves were plucked and heat killed in warm water. By heat killing, the cells become more permeable to the iodine solution. After heat killing, these leaves were placed in alcohol where the chlorophyll was removed and the leaves became colorless and brittle. To get rid of this brittle nature, the leaves were again placed in this boiling water so that now the leaves were softened. After this, both the leaves were kept in Petri plates and iodine solution was poured on it. This leaf, which is this leaf that was present or inserted inside this jar that had sodium hydroxide, could not photosynthesize, and so it shows negative test for the iodine solution. Why? Because carbon dioxide is important for photosynthesis. Without carbon dioxide, photosynthesis cannot take place. Since the sodium hydroxide had absorbed all the carbon dioxide, this green leaf could not photosynthesize and could not form starch, so it remained brown on application of iodine. But this leaf, which was inserted in a beaker containing water, had carbon dioxide present inside in the air, so it could photosynthesize in the presence of light. So it shows a positive test for the iodine solution. That is, it turns blue-black on application of iodine, proving that starch has been formed by photosynthesis. So through this entire experiment, it is pretty clear that carbon dioxide is absolutely essential for photosynthesis. Now let us perform a simple experiment to see if oxygen is liberated as a byproduct during photosynthesis. For that, take some water in a glass and take a test tube and fill it with water completely till the mouth. Insert some water plants inside the test tube and carefully insert the inverted test tube into the water glass and put a light source so that the green plant can photosynthesize. This setup is kept for 3 days. Now see, after day 2, a small bubble is formed, and at the day 3, a bigger bubble is formed at the tip of this test tube. This is the difference between day 2 and day 3. See, there is a huge bubble formation. Now, if this test tube is taken out and sealed with the finger, and then a burning splinter is introduced into this test tube, then we'll see that the burning splinter glows even more brightly when inserted into this test tube. Let us go over this experiment once again. First, we kept this green water plant in the test tube, which had been filled with water from before. Next, this test tube was kept inverted into a beaker that was filled with water. A light source was kept, so in the presence of light, this green plant photosynthesized. This setup was kept for 3 days, and at the end of day 3, a huge bubble formation is seen. Now, slowly, the test tube is taken out. It is sealed with thumb, And then a glowing splinter or a burning splinter is introduced into the test tube. And it was seen that the burning splinter glows even more brightly. Now, isn't it very clear what the gas inside might be? Yes, it has to be oxygen, because oxygen is a supporter of combustion. Because oxygen was present, this burning splinter flew and dazzled into flames. So from this experiment, it is clear that oxygen is given out as a byproduct during photosynthesis. Now, have you ever noticed banana plants? In comparison to other plants or trees of equal height, banana trees have a very limited number of leaves. But the leaf size is huge. Now, why is that? Well, had the leaf size also been very small, together with the limited number of leaves, then there would have been very less amount of chloroplast. So the rate of photosynthesis would have decreased. Now, to maintain this rate of photosynthesis, what the banana plants or the banana trees do is that they increase the size of the leaf to make up for the less number of leaves present in the banana. So, the less number of leaves in the banana is equalized by the hugeness of the leaves. Huger the leaves, more will be the chloroplast amount or the chlorophyll amount, and more will be the rate of photosynthesis.